Okay, let's start. I think that we couldn't have chosen a better way to end this session by inviting Dr. Francisco, sorry, Paco. We were told that it's not allowed to call him Francisco, Dr. Real. He's the head of the Epithelial Carcinogenesis Research Group in the Spanish National Cancer Center. He's a reference in the area of the pancreatic and urothelial cancer. The lab is aiming to identify differentiation factors and also synthetic lethal strategies. One of the interesting things, and this is what we are also trying to do here in the IDISBA, is that he has a very multidisciplinary group, and this is really something very important because each expertise adds up. And recently, they have discovered new transcription factors that help to have a better certification of patients. So with this introduction, I give the microphone to Dr. Real. Thank you very much.
So I guess that I'm allowed to take off the mask. Yeah? Okay, so first of all, thanks for the invitation. It's a pleasure to be here. It's my first plane, the first plane I've taken since February 2020. So it's a pleasure to be here, as I said. Sorry. Where is my talk? Yeah. So what I'm going to tell you is some of the work that we are currently doing in the lab on pancreatic cancer. And I've gone somewhat into the detail because I heard that there are many PhD students and postdocs here, and I guess that you don't get to, I don't know whether you have many seminars here, so I hope you'll find it interesting. 6, yeah, number 6. That's mine. I get. And one of the messages I'd like to convey, but it's very easy to convey because actually Viola just showed it, is that there's no biologist of the 21st century who is not quantitative and does not have a computational glimpse to his work. Biology without that is impossible today. So if you went into biology because you didn't want to hear about statistics, mathematics, and so on, you made the wrong choice. Sorry. I mean, it's just not possible. Okay, so for those who don't know the pancreas, this is the pancreas. Do we have a pointer? How can I point?
With the mouse.
Oh, with the mouse, okay. So this is the pancreas. It's a slide that shows the major components of the pancreas. In blue, you see the ducts, and we'll be talking about the ducts in a minute. In red, you see the islets of Langerhans, which constitute the endocrine part of the pancreas. And in black, you see the dark matter, and that's the most abundant part of the pancreas, and those are acinar cells. I hope that the animations work, and they don't seem to be working. And, okay, no, it doesn't seem that the slides are moving at all. Okay, thank you. So I have to tap first? Well, let's see. We'll try. Okay, so pancreas cancer. I'm going to show you in this slide the scenario we face in pancreatic cancer. And just to have a view of that, this is the survival of patients in whom a 2-centimeter tumor is diagnosed. So if the tumor is diagnosed in the breast or in the colon, essentially most of the patients are cured. 95% of the patients are cured. If the tumor is diagnosed in 2 organs where tumors are classically thought to be bad, such as the lung and the kidney, you can still see that the small tumor is associated with a quite good survival after 5 years, 60 to 65%. In pancreas, what I'm showing you is not the 5-year survival, it's the 2-year survival. So a 2-centimeter tumor in the pancreas is a small tumor, is diagnosed early, but early is late. Because even if you diagnose early, only 20% of the patients are alive after 5 years, after 2 years. So this is a completely different scenario of what we see in other tumors. And this is what actually has guided the principles and the focus of our work in the last few years. So why is prognosis so poor? Well, when you have a big problem, you tend to have many problems, not just one. So one of the problems is that the pancreas is located deep in the abdomen. It's not like the testis or the breast that you can feel it and do self-palpation and pick up a lump. So this is one problem. The second is that it's located in proximity to structures, anatomical structures that are vital, like the aorta and the vena cava. So you cannot make a big resection like in the colon or in the stomach. The second is that it's a very resistant, therapy-resistant tumor. It's resistant to radiotherapy, to chemotherapy, and to immunotherapy. And this may be related to the fact, as you can see in this slide, this is a very desmoplastic tumor. It has a lot of stroma, and we think that the stroma has a major impact on the biology of the disease. Another one is that patients tend to be aged. Mean age of diagnosis is 67. They have very frequently a poor general status, and 30% of the patients have cachexia at the time of diagnosis, which means that you cannot give so many drugs because the patients are too sick. Therefore, all this contributes to the scenario, and furthermore, we know relatively little about how to prevent it because we know relatively little about the causes of the tumor. So what are the causes? Well, what's very clear is that it's a multifactorial disease. We don't have a single major risk factor like tobacco that we could avoid and decrease dramatically the incidence of the tumor. And we have many different factors like family history, I'll talk a little bit about genetics later, chronic pancreatitis, diabetes, obesity, tobacco, alcohol. All of these are factors that increase the risk of developing pancreatic cancer, but maybe with an increased risk of 1.5 or at the most twofold. So none of them is major. But all of these factors are related to inflammation. And interestingly enough, we have two conditions, autoimmune disease and allergy and asthma, which are associated with protection from pancreatic cancer. And they are also inflammatory diseases. So there's something about inflammation that can be good and something about inflammation that can be bad. And we don't understand that very well. And we think that this is one of the challenges for the disease. I'm going to be talking a lot about the dark side of inflammation today. What do we know about the genetics of this tumor? Well, this is one tumor that has been very extensively studied from a genetic standpoint because it is such an important challenge from the medical standpoint. So from normal cells, and don't take this word for that here, from normal cells we know it has been reported that there are a number of preneoplastic lesions called PANINs, pancreatic intraepithelial neoplasia, and these lesions are thought to be the precursors of PANIN-3, which is essentially carcinoma in situ, and pancreatic cancer. And the main genes that are involved in this process are KRAS, which is mutated in almost 100% of pancreatic cancer, so it's almost a sine qua non condition. And it appears, according to this model that all the review papers show, it appears rather early because PANIN-1 lesions already have KRAS mutations. And it has been proposed, and this is what you'd read everywhere, that progression from PANIN-1 from PANIN-2 takes place through the occurrence of P16 inactivation, which is almost universal, 100%, and that progression from PANIN-2 to PANIN-3 and PDAC occurs through inactivation of two important tumor suppressors, P53 and SMAD4. And this is what everybody believes, and actually these four genes are almost universally altered in a pancreatic ductal adenocarcinoma. Pancreatic ductal adenocarcinoma is the most common tumor in the pancreas, and this is the one I'm going to refer to. But actually, when whole exome analysis and whole genome sequencing has been done, what has been shown that in addition to these four magnificos, I call them, there are a plethora of private mutations, and none of the other genes is altered in more than 10% of cases. So this is a very contrasting genetic scenario. But actually, I don't think that all the evidence is there. These samples that I've shown you in this graph were largely taken from patients with pancreatic cancer who had PANIN lesions in the vicinity of the tumor. And this was very much driven by the Hopkins group, who has done an amazing work. But the Hopkins groups published 3 years ago that when they looked for high-grade PANINs, so for PANIN-3s, in patients or in individuals who did not have pancreatic cancer, what they saw is that actually most of the high-grade PANINs, PANIN-3, had KRAS mutations. About 30% had P16 inactivation, but actually very few of them had P53 and SMAD4. So actually, the model that I showed you, which everybody keeps repeating, and I want to call your attention because you're young and you have to change things, is that the current model, I think, is based on the analysis of the wrong samples. Because actually, PANIN-3 is not a precursor of pancreatic cancer, it's an outcome of the tumor that progresses within the duct and looks like a precursor lesion. So we have to be very careful about how we understand these models. And we've been interested in the last few years, because of what I said earlier, in understanding the very early steps of pancreatic cancer. And these very early steps, we think they are important because they might provide us with opportunities for prevention. So which are the early events in pancreatic cancer? You cannot study this in humans. For the obvious reason I told you, we can't get easily biopsies from normal individuals or from subjects without the disease, even with people with precursors. So for these mouse models, genetic mouse models, have been absolutely pivotal. So we've made use, and we're making use very frequently, of genetic mouse models, which we try to relate to the human condition. So what have we learned? What we learned is that despite the fact that pancreatic ductal adenocarcinoma had been thought for a long time to arise from pancreatic ducts, the situation is more complex. One, acinar cells, which are the most abundant cells in the pancreas, are prone to undergo a metaplastic process that's called acinoductal metaplasia. Second, pancreatic ductal adenocarcinoma can be initiated from mutant KRAS activated in acinar cells. So acinar cells can be the source of ductal tumors. However, acinar cells can be the source of ductal tumors if KRAS is introduced during embryonic development, when they are not fully differentiated. But when KRAS is activated in mice in the adult pancreas, nothing happens. Nothing happens. No mice develop tumors. So there are other things that are required in addition to KRAS. And what we know is that in the adult, pancreatic damage sensitizes the cells to the effects of mutant KRAS. So actually, what's common between the pre-acinar cells during embryonic development, which are sensitive to KRAS, and what happens during pancreatic damage in mice that harbor a KRAS mutation activated in the adult, is that in the latter circumstance, there is a process of damage, dedifferentiation, regeneration, and redifferentiation again. So this has made us propose for a number of years that actually the first tumor suppressor mechanism in the pancreas is differentiation, doing what you have to do. If cells do what they have to do, differentiate, perform their tissue-specific functions, they don't do something else, which is cancer. That's the principle that has guided our work over the last few years. So now we know with all these genetic mouse models that actually pancreatic cancer can be initiated in mice from progenitor cells, and in this case we get PANINs and we get PDAC, from adult acinar cells and we get PANIN and we get PDAC, but it requires damage. And it can also be initiated from ductal cells at lower efficiency in a very p53 dependent manner, and it does not necessarily require damage. Okay, so it can originate from multiple cell types, but since PANINs are common in patients with pancreatic cancer, we started about 10 years ago, 12 years ago, focusing on trying to understand the very early processes of pancreas cancer development, which is this loss, we think it's this loss of differentiation, which is called ADM, or acinar ductal metaplasia. So if we want to understand how this process happens, it becomes essential to understand the nature of acinar cells and how they differentiate. Well, the first thing you have to know about acinar cells is they are the greatest protein synthesis factories in the organism. They are completely committed to making high levels of secretory enzymes, which are the digestive enzymes that digest food in the intestine. 80% of the pancreatic transcriptome is explained by 20 genes. Imagine. So they are completely committed to making these 20 genes. And what we know is that they have high capacity exocytosis. They are permanently stressed, like PhD students. ER stress and UPR are essential, okay? They require a lot of energy because they are great protein synthesis factories. They are under oxidative stress pressure. And as I will show you, this is our small contribution to the field, they suppress inflammation. So anyway, in order to understand how ACNR differentiation is deranged as a precursor of cancer, we need to understand what happens normally. My principle in the lab is that if we don't understand what happens in normal tissues, we can hardly understand what happens in cancer. Very simple. Okay, so what do we know? We know that differentiation from multipotent pancreatic progenitors to mature ACNR cells is an analog process, and there is a master regulator that's called PTF1, pancreas tissue factor 1. And in addition to this master regulator, there are a few friends that contribute to complete ACNR differentiation. The GATA proteins, the family of NR5A2 and HNF1 alpha, I'll be talking about them now, and a number of transcription factors that are involved in the control of ER stress. Okay, so we became interested a few years ago in the study of this protein, this transcription factor, NR5A2, or LRH1. It's an orphan nuclear receptor which is required for pancreatic homeostasis. And we became interested in this protein because A, it is required for pancreatic development and for full ACNR differentiation. It is known to be important in other sites in the organism, such as the liver, and in the liver it plays a role in ER stress, which is a function that's important in the pancreas. And we found that another transcription factor that's important in the liver and also in the pancreas, HNF1 alpha, regulates NR5A2. So all this brought us to become interested in NR5A2, and around the same time, the GWAS data indicated that SNPs in the vicinity, but with no functional evidence 10 years ago, in the vicinity of NR5A2 and HNF1 alpha were associated with an increased risk of developing pancreatic cancer and chronic pancreatitis. And we thought, okay, this is an interesting candidate to study. And we know also that genetic variation in the vicinity of other genes that are important for this process of ACNR differentiation are also hits in GWAS analysis. So we made the big jump that we thought, okay, these genetic variants, what they might do is, in a subtle manner, reduce the functionality of NR5A2, and the best way we could test this in mice is to use heterozygous mice. These are germline variants, therefore we decided to study NR5A2 heterozygous mice. And what happens with NR5A2 heterozygous mice? Well, you can see here the pancreas of wild type in the left and heterozygous mice in the right, completely normal. But when we submitted these cells, and this is what we do all the time in the lab, if we study a mouse and there's no phenotype that we see, we do the stress test like cardiologists, okay? So we induce damage and see whether they recover well or not. So we administer cerulean, which causes a mild pancreatitis. In wild-type mice, you can see that after 24 hours of administering cerulean, you see some edema and some damage, and by day 5, the pancreas is completely recovered. In heterozygous mice, we have more damage, and by day 5, the mice are not recovered, indicating that NR5A2 is a haploinsufficient gene for pancreatitis. And then we asked, okay, this is pancreatitis, what about cancer? So what we did is we crossed the mice so that we would have mice that carry a mutant KRAS in embryonic, I have trouble with the mouse, in embryonic progenitors, and we analyzed the development of panins and cancer in the context of wild-type NR5A2 or heterozygous, and we didn't see any difference. But when we combined NR5A2 heterozygosity, KRAS mutation, and a very mild pancreatitis, then we clearly saw that the combination resulted in an increased rate of precursor lesions and cancer in the heterozygous mice. And this is exactly what happens in patients. What happens is there's some susceptibility variants, like NR5A2 variants and other variants in other genes, a somatic mutation, KRAS, and we know that chronic pancreatitis is a risk factor for pancreatic cancer. So we thought maybe this will teach us something about how pancreatic cancer develops in the early stages. So for that, since we had not much clue about what was the mechanism, we took a simple approach, is we did RNA-seq. And we did RNA-seq in wild-type and heterozygous mice in basal conditions, again, and then after inducing pancreatitis at 8, 24, and 48 hours. So what I'm going to show you now is what happens in basal conditions in 8-week-old young heterozygous mice in comparison with wild-type mice. The pancreas looks completely normal, no leukocyte infiltration, no histological abnormalities. You do RNA-seq and you find that 1,000 genes are differentially expressed, and you do pathway analysis, and most of the upregulated genes belong to these pathways: immune response, defense response, immune system response, humoral immune response, inflammatory response, no leukocytes in the tissue. So what is this? We couldn't understand it. So we confirmed this by RT-qPCR, and actually many of the genes that were upregulated in this tissue that appeared completely normal were genes coding for chemokines or for complement components, for proteins that are involved in the very early phases of inflammation. So we confirmed that this was true, that these genes were upregulated, and we said, well, maybe NR5A2 is regulating these genes. And actually, when we looked at the presence of NR5A2 binding motifs in the promoter, simple guys, we didn't look at enhancers because we didn't have this information yet. In the promoters of these genes, we saw that they were very enriched, these motifs. 80% of the upregulated genes had NR5A2 putative binding motifs. But when we did ChIP-seq, we didn't find that NR5A2 was bound to those motifs, suggesting that there was, first of all, a discrepancy and that the effects were indirect. So then we looked at which motifs were actually enriched in addition to NR5A2, and we found the main motifs that were significantly enriched there are the ones that I'm showing you here. And we decided to focus on AP1 for a very simple reason. Sometimes scientists make simple decisions, which is that Irving Wagner, who was an AP1 specialist, and he had all the mice that you could think of in order to test whether AP1 was important, and AP1 is important in inflammation, was sitting close to us at CNIO. So we focused on AP1. And what we saw is that, first of all, AP1 components like JUNB and JUND and CFOS were upregulated in the heterozygous mouse in basal conditions, no damage, young mice, and not only that, that NR5A2 was able to bind to the promoter of CFOS and FOS1, specifically in heterozygous mice, not in wild-type mice. And then we looked at the protein level, and we saw that in heterozygous mice, which you can see here by Western blot, we see increased expression of CJUN, of JUNB, of JUND, CFOS. So clearly, there was an upregulation at the protein levels of multiple AP1 components. And then we did ChIP-qPCR with several of these AP1 components on some of the genes that I had shown you earlier that we had verified by RT-qPCR that were upregulated, like the complement components that I'm showing you here. And you can see in blue is in wild type and in red is in heterozygous. And you can see that for many of these genes and for multiple AP1 components, we saw that in heterozygous mice, there was increased binding of AP1 to the promoter of these genes, suggesting that they might be activating the expression of these genes. So what I've shown you so far is that in heterozygous mice, we have what we call a pre-inflammation, because we have increased inflammatory programs at the transcriptional level, but we don't see any leukocytes, so that's not inflammation. We call it pre-inflammation. And because it's caused by haploinsufficiency, we call it geneflammation. Okay, it's inflammation that's caused by haploinsufficiency. So what I've showed you until now is what happens in the RNA-seq in the basal conditions without any damage. But then I told you that we also checked we did a time course for the RNA-seq, and what you can see here is in the principal component, you can see in circles the wild-type mice, in triangles the heterozygous mice. And what you see is in purple at time 0, and you can see that at time 0 in wild-type and in heterozygous mice, the transcriptomes in this principal component analysis are different. But actually, at time 0, the transcriptome of heterozygous mice, the red triangles, sits essentially on top of the transcriptome of the 8-hour pancreatitis in wild-type mice. So actually, this confirms that what is happening in these mice in basal conditions is that it's almost as if they were entering a pancreatitis, but they don't continue, they don't go on. So this is the pre-inflammation. And then we check, well, we know that NR5A2 main function in the pancreas is to regulate ACNR genes. So let's look at what happens during the inflammation induced by pancreatitis with the distribution of NR5A2 in the pancreas. And you can see that when we look at the ACNR enzymes, the ones coding for digestive enzymes, at time 0, which is the first time point, we have binding of NR5A2 to their promoter for multiple of them, and then upon administration of cerulein, very rapidly, within half an hour and 1 hour, we see that NR5A2 appears to be displaced from these promoters, which are characteristic of the tissue-specific program of the pancreas. And then we looked at NR5A2 in the inflammatory genes. And as you can see, in basal conditions, at time 0, NR5A2 does not sit on the promoter of those genes, but actually, after administration of cerulein, you see that NR5A2 binds to the promoter. And indeed, this is also true for AP1. So what this means is that I'm going to show you this in a heat map because it's a little bit easier to see. In the left side, you see this is basal conditions, young wild type or young heterozygous mice. In red, you see a whole bunch of ACNR digestive enzymes, so the tissue-specific genes. In orange, you see the AP1 proteins, and in blue, you see the inflammatory genes that are upregulated in these mice. And you see that in wild-type mice, NR5A2 binds mainly to the tissue-specific program genes. By contrast, in heterozygous mice, there is downregulation of binding to these tissue-specific genes and upregulation of binding to inflammatory genes. And if you compare this with what I'm showing you here, which is the time course of distribution of NR5A2 in this set of genes after initiation of a pancreatitis, you see that in wild-type mice, NR5A2 is first bound to the ACNR genes, where it should be. You induce a mild pancreatitis, and it goes away from the pancreas specific genes to the inflammatory genes, and during recovery, it goes back to the pancreatic genes. So actually, this is what we call the NR5A2 transcriptional switch. A given transcription factor does one function in a certain set of conditions and can do something completely different when something happens to the tissue. And the next question was whether this was actually truly due to AP1, to c-Jun, and what we did is, because we had Irving there and he gave us all the mice, we deleted, here you can see in the second panel, induction of pancreatitis in a second panel, in a heterozygous mice that was c-Jun competent, and compare with a heterozygous mice that is c-Jun null. And you see that deletion of c-Jun completely rescues the effect of NR5A2 heterozygosity, proving that AP1 is the mechanism that mediates the sensitivity to pancreatitis in the heterozygous mice. So this is the model. In basal conditions, as I said, I'm going to skip this or go through this rapidly. In basal conditions, NR5A2 mainly binds to the ACNR genes. Upon damage, or in heterozygous mice, it binds to AP1 and to inflammatory genes. So then the question is, once we have this pre-inflammatory state, who and how is this pre-inflammatory state established or maintained? So we've done 3 types of experimental manipulations, and I'm going to refer to one of them today. Aging, we didn't see much with aging. Metabolic stress, we've seen things, but I'm not going to talk about them today. But we looked at the microbiome, and we asked, is it possible that actually the pre-inflammatory state of the pancreas depends on what's going on with the gut microbiome? So what we did is we took wild-type mice or heterozygous mice. We gave them an antibiotic cocktail for a week. Then we kept them on ampicillin to suppress bacterial growth over 3 weeks, and then we induced a pancreatitis. And what you can see that in mice that drank water, of course, heterozygous mice, as I told you earlier, have a more severe pancreatitis than wild-type mice. But if we deplete the gut microbiome, and I'm not going to show you evidence for that, but I'm telling you that we clearly depleted, the sensitization to pancreatitis that we had found in heterozygous mice completely goes away. The mice actually respond even better than the wild-type mice. And we can show this here when we look at the histological scores. We can show this here when we look at amylase. This is the image of what happens in the heterozygous mice drinking regular water. And this is what you see, the increase in serum amylase in wild-type versus heterozygous mice, much higher increase in serum amylase because the pancreatitis is more severe. And when we give antibiotics, nothing happens in the wild-type mice, essentially the same. But when you look at the heterozygous mice, there is complete rescue of the aggravation of the pancreatitis. Okay, so let me see if this moves. Now it doesn't want to move. Okay, so then how does this happen? So in order to get clues about how this happens, one these days takes omics approaches, metabolomic or genomics or whatever. We have begun to do the metabolomics approaches, but I'm showing you here the RNA-seq analysis. And what we saw is the following. First, we did RNA-seq, in these various conditions, wild-type or heterozygous mice receiving water, and then, or antibiotics, and then being analyzed in basal conditions or after pancreatitis. And what you can see is that if we compare pancreatitis versus basal conditions, as we expected, because we've shown that in the past, upon induction of pancreatitis, both in wild-type and in heterozygous mice, we see a down-regulation of the ACNR program because cells de-differentiate, and up-regulation of inflammatory pathways, immune, inflammatory, leukocyte, chemotaxis, etc. What happens when we compare antibiotics versus water? Well, you see in light red the wild type and in darker red the heterozygous. So in basal conditions, actually the administration of antibiotics increases the activity of the normal pathways present in the pancreas, the ACNR gene program. And when we look at pancreatitis, we see that there is actually an increased activity of these pathways, which would be normally downregulated because of the damage, but this effect is greater in the heterozygous than in the wild-type mice, in agreement with the fact that histologically we saw rescue in the heterozygous mice, but we didn't see much effect in the wild-type mice. So what happens regarding the anti-inflammatory effect? Well, what you can see here is that in wild-type and in heterozygous mice, in basal conditions, the administration of antibiotic reduces inflammatory signatures in the pancreas to a similar extent. But upon induction of pancreatitis, the only strain in which antibiotics rescue the inflammatory program is in the heterozygous mice, but not in wild-type mice. So actually what we are seeing is a microbiome, we think, mediated. Now we have more evidence because if we rescue the gut microbiome, we rescue the phenotype, but we see a microbiome-dependent effect that is specific for a given genetic context, heterozygous mice. So we think that obviously this raises the possibility that one might be able to manipulate, not by depleting the gut microbiome with antibiotics, but in other manners, by manipulating pharmacologically or with diet, the behavior of the lifestyle factors, we might compensate for what appears to be an insurmountable problem, which is the genes you inherit from your parents. This is the same thing that we have seen when we look at many pathways. You can see, just focus on these two graphs here. You can see that when we give pancreatitis, when we administer or induce a pancreatitis in the wild-type mice, in the heterozygous mice, and there's a mistake here because this is without and this is with antibiotics, we see, a clear reduction in the activity of the inflammatory pathways and a clear reduction in the activity of the cell cycle pathways. Anyway, what we're doing now is we're checking which are the putative transcription factors that might be regulating this, and one of the main suspects is PU1. You heard about PU1 from Viola. PU1 is very important in neutrophils and in monocytes. And we have some evidence now that actually the administration of antibiotics to the mice, I'm not showing this here because it's rather complex, does have an impact both on the circulating monocytes as well as in the tissue-resident monocytes, and that these may be the cells that are the culprit for the effects of the gut microbiome depletion. Okay, so as I told you, we know now that the differentiation process in the pancreas is a complex process that involves many partners. So the next question we asked, in parallel actually, was how is the NR5A2 haploinsufficiency affecting the function of NR5A2? And we took a relatively simple approach. We said, well, what happens with the interactome of NR5A2 when you reduce the dose of NR5A2 by half? And this was an amazing experiment for me. What you see here is in the first column, in the first column of the heat map, okay, you see the binders of NR5A2 when we do an immunoprecipitation and mass spectrometry in basal conditions. What you see in the second one is what happens in basal conditions in heterozygous mice, the binders. And what you see in the third is the wild-type mice after 1 hour of administering cerulean, inducing a pancreatitis. So actually, what you see here is that the heterozygosity of NR5A2 results in a complete shift in the interactome of a transcription factor, reducing the dose of a transcription factor by 50%, dramatically shifts the interactome of this protein, and this is much more similar to what occurs during pancreatitis than to what occurs in wild-type mice in basal conditions. I was struck about this finding. So haploinsufficiency, which is something that we don't think about as something very important in genetics normally, can have profound biological effects at the cell and tissue levels. And indeed, through this approach, we identified a new transcription factor. I'm going to go through this quickly. I don't want to bore you with this, called NFIC, which was not known at all to be involved in the pancreas, and which, through this strategy, we uncovered that it binds NR5A2 in co-IP and Western experiment, and it is important for acinar differentiation. So NR5A2 is expressed in, NFIC is a ubiquitous protein, is expressed in acinar cells, but not in ductal cells, it binds to the promoter of multiple genes coding for acinar enzymes, like its friend and interactor NR5A2. It is regulated by NR5A2 and by other important transcription factors because this is a gang where there is a lot of cross-regulation. And actually, when we knock down NFIC in acinar cells in vitro, we do see a downregulation of both, transcription factors that are important for pancreas differentiation. I don't like these mice. Sorry. I like my mice. I don't like these mice. There's a downregulation both of transcription factors and of the acinar enzymes. So NFIC is, we've uncovered that it's an overregulator of the acinar program, but it does things slightly different from what NR5A2 does. NR5A2 controls the acinar program and suppresses inflammation. NFIC, actually, its major function appears to be to regulate ribosomal gene activity and the ER stress and UPR. And the message here is very simple. We have a whole gang of transcription factors that cooperate to make a cell do what it has to do, and each of them has a little bit of a specialization in order to provide a module, this is what we now have evidence for, of that differentiation program. In the case of NFIC, is suppression of inflammation and acinar differentiation. In the case of NR5A2, in the case of NFIC, is acinar differentiation and regulation of the ER stress response. And you can see this here because we see that when NFIC is knocked out in the pancreas, we have the differential activity of a number of pathways related to ribosome biogenesis. We see reduced 5-S ribosomal RNA here by immunofluorescence, and we see that indeed NR5A2 and NFIC bind to multiple ribosomal proteins, and they also bind to proteins involved in the UPR, in the activation of the stress response, which is a response that is crucial for the viability of acinar cells. I'm going to skip this. So, if I summarize this, what we know that for acinar cells to do what they have to do, they have transcription factors that are involved particularly in the activation of the exocytosis machinery, like MIST1. Those that are involved mainly in ER stress, and I showed you NFIC. Those that are involved mainly in suppression of inflammation, like NR5A2. And these different components of the cellular function are driven by different transcription factors that cooperate to make the whole program work. But then the question is this any relevant to humans? Because I started by telling you that NR5A2 genetic variants were associated with humans. So what we did is we've done a number of things. But this is a summary of them. What we've done here is we've taken the 1,000 genes that were upregulated in the NR5A2 heterozygous mouse, and we took GTEx normal pancreas data from individuals that are completely normal, and we took the highest quartile of NR5A2 expression and the lowest quartile of NR5A2 expression. And we compared the expression of these 1,000 genes that were upregulated in the heterozygous mice in the low versus high. And as you can see here in this quadrant here, 80% of the genes that we found that were upregulated in heterozygous mice are upregulated in the pancreas, in the normal pancreas of organ donors that have low levels of NR5A2. So we think that this is relevant to human, and something similar we've done for NFIC, and we see exactly the same thing. So we think this is relevant for human. We don't have final evidence that, as Viola showed, these SNPs, either in enhancers or in the vicinity of the promoters or within the gene, are actually responsible for this. But NR5A2 levels are important. So in summary, the message is that lineage-specific transcription factors coordinately control differentiation and inflammation in the pancreas. In normal conditions, the tissue-specific programs are up and inflammation is suppressed, but this is done in a coordinated manner. It's not two different programs. It's the same transcription factor, NR5A2, that does both. In pre-inflammation, we see an upregulation of inflammatory components in the absence of leukocytes. In inflammation, in pancreatitis, we see a full-blown inflammatory reaction. And the first state is associated with mutant KRAS resistance, which is what happens in the pancreas of adult wild-type NR5A2 mice. And the second state is associated with mutant KRAS sensitivity, which is what we see when we induce pancreatitis or we delete one allele of NR5A2. And where we're going from here, we're going, and this, I come back to the first, one of the first things that I said, we need to look at these processes in a more global manner. So we're trying to understand and to reconstruct the transcription factor networks that are involved in this process. It's not one transcription factor, as I told you, it's a gang. And we need to understand who are the members of the gang and how each one of them contributes to this. We are integrating ATAC-seq and ChIP-seq data to identify regions of open chromatin, looking at footprinting to actually identify which regions are being occupied by transcription factor, generate transcription factor binding maps, and then look at individual and global networks, integrate this with, RNA-seq bulk and single-cell data, look into functional validation, and eventually link this to the GWAS analysis so that we can identify better individuals who might be at increased risk of pancreatic cancer, and maybe through the analysis of the microbiome studies or through other studies, we can identify pharmacological manners to prevent the development of the cancer, because once it's diagnosed, it's too late. Okay, so just to finish, some concluding remarks. Inflammation plays a dynamic role in cancer very early, and late, at all stages of cancer development. Epithelial differentiation is coupled to the suppression of inflammation. Pharmacological modulation of differentiation and inflammation may be used to suppress cancer development. And I think we need to integrate, you young guys, need to integrate computational and experimental analysis. That's exactly what Viola is doing, to discover new genes involved both in normal conditions and in cancer. I didn't do anything of this. So the guys who did this, the NR5A2 work was done by Irene Cobo and by Irene Millán. The bioinformatician, the main bioinformatician in our lab is Jaime Villarreal, who's been great, and with Pablo Perez, they're doing all the network reconstruction that I have told you. Much of the work has been done in collaboration with Nuria Malats, who is a genetic epidemiologist at CNIO, and with Irving Wagner. And and I'm very lucky that I have two people that are related to Mallorca in our lab who are absolutely outstanding. Auba Gaya, who is a PhD student, who is trying to unravel why asthma protects from pancreatic cancer, which is, I think, a fascinating question, and Irene Felipe, who did her postdoc here at Son Espases. And that's all. These are the funders. Thank you. I'll take questions if you have any.
let's have a nice discussion of this fantastic talk. Please, questions? Oh, what? Up.
I discovered the mouse. Too late.
No, it's not well placed, though.
Yeah, it's difficult.
Hello. It's great to have you here, Paco. So it's really inspiring and interesting research. And my question is related to the geneflammation of NR5A2. So perhaps I miss it, but is it somehow involved to the stem cell differentiation state? Somehow, I mean, is LGR5 or well, you mentioned GETA-CIDS program. Is it involved in this step?
Well, what I can tell you is that from what we know is that in the differentiated cells, NR5A2 is suppressing inflammation. Is actually activating the differentiation program. I mean, we're looking at it from the opposite side of stem cells. The stem cells are the ones that are not differentiated, and we're looking at the other side of the coin, the differentiated cells. So it's rather similar. What we can say is that NR5A2 in differentiated cells, it's both promoting differentiation and at the same time suppressing inflammation, because when we remove one allele, the inflammatory programs go up. We think that this is relevant to cancer because actually we've done a number of bioinformatics studies with TCGA data looking at breast cancer, at pancreas cancer, and at bladder cancer. And what we see is that the tumors that lose their cell lineage identity, for example, in the breast, the tumors that acquire basalness, or in the bladder, the tumors that acquire basalness, those tumors are more inflamed than the tumors that maintain urothelial or luminal breast differentiation. And those who work on breast cancer here know that. The luminal tumors are much less infiltrated in the breast than the basal tumors. So we think that this is relevant also to cancer, and this may be a more general, not the more general, that more transcription factors in addition to NR5A2 may be doing this.
Okay. And my second question is related to microbiota. I mean, it has been largely described that microbiota diversity has an impact on tumor progression, so that the less biodiversity you have, the more the tumor is promoted. So, have you had a look at that? And,
yeah.
And sorry, and another question. This is from your studies from gut microbiota. Have you looked at intratumoral microbiota, which is another?
Yeah, we've looked at, I mean, most of the experiments that we do with the wild type and the heterozygous mice, the mice are co-housed. So we didn't expect to see differences in the microbiota. However, because this was a question that the reviewers will ask, even if we didn't expect it, we have done a microbiome analysis using 16 sRNA, and we have not seen significant differences between the wild type and the heterozygous. But what we have done also is, after depleting the microbiota, I haven't shown this data, it's quite recent, after depleting the microbiota, because one could say, well, maybe this is an effect of the antibiotics, not of the microbiota, we've reconstituted the microbiota not through fecal transplantation, because we cannot do that at CNIO. The animal room is too clean to do those things. But we've co-housed the heterozygous with the wild type, or the heterozygous with the heterozygous that had not been treated with antibiota, and then we completely rescue the phenotype. So it is definitely microbiota driven. We cannot identify any differences, and we don't know exactly where the cause might be, to be honest with you. It may be bacterial load. We're trying to look at whether it is load in general of bacteria that might be the cause. But we haven't seen any species-specific difference, and we haven't looked at tumors, no. Nuria Malatz, who is our collaborator, has been looking at human data, and actually has recently has a paper submitted, I think it's in bioRxiv, where she shows that there are specific microbiota species that can be identified in subjects with early pancreatic cancer and that may have diagnostic use, but we haven't looked at the tumor itself.
Okay, thank you.
Any other question?
I knocked you out? I thought it was going to be only haploinsufficiency.
From the remote?
I have Dr. Real, thank you so much for your presentation and for coming
You're
to
welcome.
join us.
My pleasure.
My question was exactly very similar to what you just answered about the microbiome transplantation. My question was if you were able to transplant microbiome and rescue the phenotype. But
Well,
I would like to emphasize from your talk how important is to consider the genomic context, the epigenomic context, and now the microbiome context for mutations to really have activity in the phenotype.
Well, yeah, to me this is fascinating. The first thing that to me is fascinating is that we can manipulate something. I mean, I usually say the only risk factors that you cannot get rid of regarding cancer prevention are your genes. Sorry, your parents gave you these genes, that's it. But now I think that you cannot get rid of your genes, but you can do something about it. So obviously, as I said, we cannot get rid of the microbiome, but if we are able to unravel how the microbiome acts, then we may be able to find some ways to do more therapeutic prevention in high-risk individuals.
Amazing.
I had another question. Did you do the metabolomics? I mean, I'm thinking about the inflammatory molecules and our signature and how it changes. Because if you think that inflammation is involved and it causes such a damage from the very beginning, if they have a different signature, you know, I'm thinking about all the prostaglandins. Well, there are many more, and now there are all these pro-resolvings and all. If you have analyzed this,
no,
signature.
actually, I think what you just raised is a very interesting thing. I think that there's a lot of information about I mean, here what we see is that we see more damage, and we see slow recovery. But we don't know whether the slow recovery or the worse recovery is simply due to the fact that there is more damage, or because some of the resolvins or other lipid mediators that are involved in resolution of inflammation might be affected. I think this is a very good idea. We haven't done it yet, and I think it would be worthwhile doing, yeah, definitely. Because affecting the resolution phase of inflammation might be equally important or more important than just damage.
Curiosity, the link between the gut microbiota and the pancreas? I mean...
Well, the pancreas is very close to the duodenum. I mean, the duct ends up in the duodenum. Mm-hmm. We know, I mean, if you had asked me 10 years ago, I would have said, well, but the duodenum, or you would have said, yes, but the duodenum is sterile. But nothing is sterile anymore. No. We know that nothing is sterile. So we know that there are bugs in the pancreas, low biomass, okay, but there are some bugs that can be detected in the pancreas. It is absolutely conceivable that from the duodenum, where there are microbiota, they could enter through, and there is some evidence actually, that they could enter through the pancreatic duct into the gland. So I think that it is completely conceivable to me that there could be a local effect. We have not looked at the duodenal microbiota composition in wild-type heterozygous mice, which is something that I know it's a flaw of the study, because NR5A2 could be affecting the intestine rather than only the pancreas. These are germline variants, so this is certainly possible. We haven't looked at that yet.
If there are no any other questions, we will close this session with a big applause to our last speaker.
Thank you.
And